Hello, I'm Dr. Mark Darcy, and in today's session I'm going to be discussing DNA. I'm going to tell you what DNA is, who discovered the structure of DNA. I'm going to be talking about how DNA gives rise to something called RNA, and how RNA gives rise to the proteins. So I'm basically going to be covering the topics of DNA replication and gene expression. We have 25,000 genes in our body approximately, and most of those genes carry the instructions to make proteins. Most of what we are proteins. So the insulin that circulates our blood is a protein. The collagen in our skin is a protein. The keratin in our hair is a protein, and our digestive enzymes are proteins. So we are protein, and those proteins are coded for by genes in our DNA. Okay, so let's get started. Okay, so DNA replication and gene expression. So let's begin with DNA makes DNA. How we replicate our DNA and the structure of DNA. So DNA makes RNA makes protein. This is known as the central dogma of molecular biology. DNA makes DNA is known as replication. DNA makes RNA is known as transcription, and RNA makes protein is known as translation. Francis Crick was the person who proposed this central dogma of molecular biology back in 1958, and it was slightly reworded in 1970, but basically said the same thing. You may have heard of Francis Crick. Together with James Watson and Rosalind Franklin, they managed to ascertain the structure of DNA. Okay, so nucleic acids. What are nucleic acids? So nucleic acids are basically what DNA and RNA are made up of. That's what they are. DNA is always double-stranded except in some viruses. So you get two strands of DNA held together. RNA is always single-stranded, except in some viruses. And there are three main types of RNA. Messenger RNA, which carries the message, the instructions from a gene in the DNA to a structure called the ribosome in the cytoplasm that makes a protein. So we have 25,000 genes approximately. A gene is a piece of our DNA on our chromosome that carries instructions to make some protein. Those instructions are carried to a machine in the cytoplasm called a ribosome, which reads the instructions and makes the desired protein. And they are carried there by messenger RNA. I will get to ribosomal RNA shortly. That's basically what the ribosome is made of. It's made of a type of RNA. And I'll tell you about transfer RNA later as well. Basically, transfer RNA is what helps assemble the protein at the site of a ribosome. Okay, so here, as you can see, we have DNA and RNA. So DNA is double-stranded. It's double-stranded. A double helix, I'm sure you've heard that term before. The helix contains these molecules: cytosine, guanine, adenine, and thymine. Adenine always binds with thymine, AT. Cytosine always binds with guanine, CG. So these little arms that you're seeing coming out from the DNA, they are the bases: the adenine, the thymine, the guanine, and the cytosine. A binding with T, C binding with G, all the way along. And that sequence of A's, T's, C's, and G's is your genome. And several thousand of those together might make up one gene, the instructions to make one protein. And as I said before, we have about 25,000 of those. You might wonder what the backbone of DNA is made up of. This backbone is made up of phosphate and sugar. DNA, the sugar that makes up DNA, is known as deoxyribose, and that's linked with phosphate. So it would be phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way along with these bases, adenine, thymine, guanine, and cytosine, sticking out. So this is DNA, deoxyribonucleic acid. Here is RNA, which in all eukaryotic cells and our bodies are made of eukaryotic cells, is single-stranded. It's still the same here, sugar and phosphate, the difference being that the sugar is not deoxyribose, it is ribose. Ribose phosphate all the way along, but just with one base sticking out, not pairs of bases, but single bases. The other difference is that instead of a thymine in RNA, you have a uracil, so it's AU instead of AT. That's the key difference. Okay, zooming out. Before I go and talk about RNA, tRNA, before I discuss messenger RNA, transfer RNA, and ribosomal RNA, I just want to go through a little bit more about the structure. So, organization of the genetic material in cells. In bacteria, the genomes are covalent, closed, circular DNA. So there's one chromosome, one big chromosome. The thump you just heard was my cat jumping onto the table. Please ignore that. So the DNA that we have in prokaryotic cells, that means bacteria, is circular. There's just a circular piece of DNA, circular chromosome, which floats around in all prokaryotic cells. It's not bound by a nuclear membrane like eukaryotic DNA is. It's just a circular piece of DNA. CCC, covalent, closed, circular. In eukaryotic cells, such as us and plants and fungi, the chromosomes are linear DNA packaged in proteins, histones. So the DNA is wrapped around these little balls called histone proteins and then condensed into structures known as chromosomes. And we have 46 chromosomes in our cells in a nuclear membrane, so they are trapped in a nucleus. So they are linear rather than circular in eukaryotes. They are so condensed in the form of chromosomes in eukaryotes because we have so much more DNA than the prokaryotes, about 2 meters of DNA in each one of our cells, in fact. In archaea, which is a sort of primitive eukaryotic cell, the genomes are CCC, but the DNA is packaged in histones, so you can see there's sort of an in-between species between the prokaryotes, bacteria and algae, and the eukaryotes, animals, plants, and fungi. In bacteria and some archaea, genomes are condensed into a tightly called structure called the nucleoid by various proteins. Okay, before I get into the DNA and tell you how it replicates, I just want to mention a few key players. So first of all, Watson and Crick. Here they are in a very posed-looking photograph. This was the guys when they just worked out the structure of DNA. This was before computers. This was before any sort of modern kind of technology. They worked all of this out by putting models together and by looking at a particular image generated by X-ray crystallography. This is a technique whereby you can look at very, very small molecules that you cannot see with a light microscope. Now, X-ray crystallography is very difficult, very hard to get right. Before these guys came up with the structure of DNA, we were pretty sure that DNA was the thing that was passed from generation to generation and was responsible for our traits and inheritance. But we didn't know the structure of DNA. So without knowing the structure, it was hard to know how this stuff worked, how it passed information along. So it was key to work out the structure. And these guys did it. But they couldn't have done it without the help of this woman here, Rosalind Franklin. Now, Rosalind Franklin was a PhD student. I believe it was at UCL. I could be getting that wrong, but it could be Imperial, but I think it was UCL. And she was working on a PhD thesis, and she was performing X-ray crystallography. She was very, very good at it. And she had taken an X-ray crystallography image of DNA. You don't get the full details from an X-ray crystallography image, but she got a kind of cross image on a photographic film, which gave a clue towards the structure of DNA. Now, without her permission, her supervisor happened to know, a PhD supervisor happened to know Watson and Crick, passed on her image to those guys, and they used that image to help them to work out the structure of DNA. Now, it wasn't as simple as that. There was still a lot more work to do, and they certainly deserved the Nobel Prize that they got for discovering the structure of DNA. But most people now agree that who also deserved the Nobel Prize was Rosalind Franklin. But she didn't. She didn't get the Nobel Prize, and she didn't really get any recognition for the discovery of the structure of DNA until years and years later. But really, the structure of DNA was worked out by Watson, Crick, and definitely Rosalind Franklin. Okay, so now let's talk a little bit more about how DNA is replicated. So what we have here is we have prokaryotic DNA. That's the DNA as you see it in a bacteria or an algae. It's circular, CCC. So you can see circular DNA. In eukaryotic cells, however, such as us, in our cells, the DNA is coiled and coiled and coiled and coiled, tighter and tighter until you get these chromosomes, 23 pairs of chromosomes. So it's a very different structure. It's a very different structure. Now, I'm going to get to this in a second, how DNA is replicated. But before I do so, let's just zoom in here at the structure. So in eukaryotes and in some archaea, the basic chromosomal unit is known as the nucleosome. Nucleosomes are complexes of DNA wrapped around histone proteins. So kind of like string around balls. The histones are arranged in optimers, so it's not really one ball, it's 8 balls all together with DNA wrapped around them. DNA is coiled around these molecules about 1.65 turns per optimer, and then they are further clustered and tightened up to form chromosomes. This is what they look like. Here we go. So as you can see, we have the ball of histone proteins with DNA wrapped around. Then it wraps around the next ball of histones and the next. And then the whole thing coils together and coils together again and again and again to make the chromosomes. Okay, so now we should really talk about this, how DNA is replicated. Well, first of all, the DNA itself needs to be unraveled from the histone proteins in eukaryotes. It doesn't in prokaryotes because there are no histone proteins. So as a result, DNA replication in prokaryotic cells is much quicker than in eukaryotic cells. Plus, there's less DNA to replicate because there's less of it in a prokaryote than there is in a eukaryote. Okay, so zooming right in on these images. What are we looking at? So we're looking at DNA. I know it's normally a double helix, but we're kind of straightening it out here to make it easier to see. So let me just zoom out slightly so we can get the full image. Whoops, sorry. Just want to get a good, clear image of this. Okay, this will probably do. Okay, so what we're looking at here, we have DNA. So what we're seeing here are 3s and 5s. So DNA is what we call antiparallel. The 2 strands of DNA go in opposite directions. One is 3 to 5, the other is 5 to 3. The 5 and the 3, I'm not going to go into the details of that right now. That is a bit more biochemistry and molecular biology than we need right now. But long story short, the backbone of the DNA is a sugar, a phosphate, and a phosphate, as I mentioned. Now, the sugar has a number of carbons around it. It's a carbon ring. And each carbon has a number. Carbon 1, 2, 3, 4, 5. Now, as we see here, this just means 3', the 3 carbon end of the sugar. This is 5', the 5 carbon end. So 3, 5, 3, 5, 3, 5, 3, 5 is the direction. Now, the thing is, the enzymes that are involved in DNA replication, these enzymes, they can only move in one direction. So what's happened here is DNA is unwound. It's unzipped. It's opened up. So you're seeing one single strand here in red and another single strand here also in red. This is opened up, replication bubble. Now, what happens first when this bubble opens is an enzyme which is known as primase. Primase puts this green little thing here called a primer onto the single strands. So we'll start with this side, then we'll talk about this side. This is the leading strand. I'll explain what that means shortly. So we have a primer here, a primer. The reason we have a primer is because the enzyme that replicates DNA cannot bind to a single strand of DNA. It needs to bind to a piece of double strand, but the DNA is open, so it's single-stranded. This primer has bases, just like DNA does. These little bases, remember adenine, thymine, guanine, cytosine, and uracil for RNA. This is made of RNA, this primer. So the primase puts its primer here, which will bind to a piece of DNA. It binds in a region known as the promoter. The promoter. The promoter is a part of the DNA that can open up easily, and a primer can be inserted onto. So this primer binds, and when this primer binds, then what can happen is an enzyme known as DNA polymerase. So DNA polymerase, you can guess what that means. DNA, it involves DNA. It's a polymerase. It makes a polymer. Anything with ACE is an enzyme. So it's an enzyme that makes a polymer of DNA, meaning it makes DNA. So this DNA polymerase will walk along the single strand in this direction. It can only go in one direction, this direction. That's why, because the DNA is anti-parallel, this strand is in an opposite orientation than this, the DNA polymerase has to walk along this way on the leading strand, but on what we call the lagging strand, it has to walk this way. Anyway, there are a number of enzymes involved here. There are these things called transcription factors, which sort of bind to DNA when it's time for the DNA to be replicated. Sorry, not transcription factors. The transcription factors are involved in the transcription of DNA to RNA, which I'll talk about later. But what we have here is we have a number of enzymes, such as DNA A, DNA B, DNA C in prokaryotic cells, and they bind to a particular region of the prokaryotic DNA, we'll do prokaryotic first, known as an origin, for origin of replication, ORI. Specifically, if we say look at E. coli, the site is known as ORI-C. So imagine there's a region here called the origin, ORI-C. Now this is where the DNA will open up to start with, and where the primase will put the primer. Then places the primer, and DNA polymerase will walk along. Now, DNA polymerase needs a few other enzymes to help it along. So DNA A, B, and C help to open up the DNA in prokaryotic cells. But then you need an enzyme called DNA gyrase to sort of keep the DNA open, because the DNA wants to bind again. Those adenine, thymine, guanine, and cytosines want to bind together. So the gyrase kind of keeps it open. And then another enzyme called helicase moves along and sort of unzips the DNA ahead. So the DNA will be unzipped all the way around. All the way around. So this primer is bound, DNA polymerase is bound. It's now walking along, copying, with the helicase unzipping the DNA around it, goes all the way around, and it looks at this strand, and it puts the correct opposite base in. So if it sees an A, it puts a T. If it sees a C, it puts a G, etc., all the way around. Until eventually it gets all the way around to another site known as a ter sequence. Ter means terminal. That's where it stops. If there wasn't a ter sequence, then it would keep going around again and again and again and keep making more and more copies of DNA. If it doesn't, it stops at the ter sequence, and then that one circular piece of DNA will divide. You'll have two circular pieces of DNA, which can go to opposite ends of the cell. The cell can divide in two. You have two daughter cells. But wait, we've missed this strand. Now, because the DNA is opening up in one direction, and DNA polymerase can only move in one direction, it can only move this way, which means on the anti-parallel strand can only move this way, 3, 5, 5, 3, then what we have to do is, as it unzips, we have to keep placing primers along and along, along and along each primer. And then the polymerase will walk in little small chunks from primer to primer. Each of these pieces of DNA between the primers on what we call the lagging strand are known as Okazaki fragments. Okazaki fragments. And this mess needs to be cleaned up when the DNA is replicated by other types of DNA polymerase, which I'm not going to go into right now. So, end of result is the polymerase has gone all the way around from origin site to ter site, and you've doubled the amount of DNA you've got. You've now got 2 circular pieces of DNA. The cell can divide into 2 daughter cells. In eukaryotes, it's a little bit difficult. A little bit different, because we have these linear, straight chromosomes, not a circular piece of DNA. So you can see an obvious problem. You can't put a primer just in mid-air. It has to be attached to something. It has to be on something. Here, the primer is on the DNA. And on the opposite side, here the primer is on that side of the DNA too. So that's no problem, because DNA will replicate all the way around, but the bit with the primer on can't be replicated. It can't be replicated in one direction. This is where the primer is, this is where the polymerase is, which goes that way. So that bit doesn't get replicated at the end. These little bits in the middle can be replicated because you've got double-stranded DNA around it, and you've got a special kind of polymerase which can go in and clean that up. That's not a problem with prokaryotes, because it will go all the way around the polymerase and eventually get back here, so it can clear the mess up. But with linear, straight chromosomes, that can't happen because there is an end to the chromosome. Remember, the end is where the primer will be, so it can't be copied, the DNA next to that primer, which basically means every time a linear chromosome, such as the ones we have in our cells, all eukaryotes have, every time it replicates, our chromosomes get a little bit shorter. You may ask yourself, then what happens to our DNA? Surely our chromosomes, if they get shorter, we lose genes. Well, luckily, there is an answer to that. The very ends of our chromosomes have a type of DNA on them known as a telomere. Think about telomeres at the end of your chromosomes, kind of like the plastic on the end of a shoelace. It's there to protect the shoelace from fraying. And that's exactly the case with the telomeres at the end of our chromosomes. Every time a chromosome gets shorter, you lose a bit of junk DNA. By junk DNA, I mean the DNA in the telomeres. It doesn't make any genes, that DNA, just there to protect the chromosomes. So chromosomes get shorter each time in eukaryotic cells. But they can only get so short before you run out of telomere, and then you're destroying genes. So in eukaryotic cells, our cells are programmed to only be able to divide a certain number of times. Between about 30 and 40 times, it's known as the Hayflick limit, from the person who discovered this limit, a scientist called Hayflick, unsurprisingly. So cells will divide about 30 or 40 times before the telomeres run out. Then they will either go into a process called apoptosis, which is when a cell basically commits suicide, or they will sit there, no longer dividing, and they will just pretty much do nothing in your tissues. They will accumulate in your liver, in your brain, in your skin, in other parts of your body, and they won't really do much except secrete bad chemicals that cause inflammation. Aged tissues, an aged heart, an aged liver, an aged kidney is full of these cells. They're known as senescent cells. They're quite damaging. They accumulate with age. They're actually helpful in small numbers. They help with wound healing. But as you get older, they accumulate and they can cause harm. I talk about that in another talk of mine. Okay, so that's what telomeres are for. I won't talk at length about that now. So that's how we replicate our DNA. DNA replication, it involves a site known as an origin, which opens up. There are multiple origins, by the way, along chromosomes. the origins open up a bit of DNA, and then within that DNA, at the origin site, I think I confused things a little bit. I mentioned promoter. Promoters are more for transcription, which I will be talking about later. But at the origin site here, the DNA opens up. We use enzymes such as DNA A, DNA B, DNA C to open it up. Then the primase will place a little primer to make a piece of a single-stranded DNA double-stranded. The DNA polymerase will bind to the primer, walk along. We will have gyrase, keeping the DNA open, helicase, unzipping it all the way around. And then DNA polymerase will walk along, turning single strand into double strand until you end up with the two separating, and you've got two new circular pieces of DNA, or single linear DNA, known as chromosomes in eukaryotic cells. These are the Okazaki fragments on the lagging strands, primers in between them, because DNA polymerase has to replicate this way on the lagging strand in little chunks, and then it's all tidied up by various enzymes, and we've replicated our DNA. Okay, so moving back and moving back again. I believe we've talked about the structure of DNA. We talked a little earlier here about the structure of DNA. So now we're going to talk about the RNAs. So first of all, messenger RNA. This is messenger RNA. Messenger RNA is single-stranded. The basis that it has adenine uracil instead of thymine, guanine, and cytosine. And this is a sort of close-up. This is the sugar, ribose. This is the phosphate group, sugar. And then sticking out from the side, we have the base, sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way along. Here is tRNA. Now, before I talk about tRNA, I need to tell you what messenger RNA is for. I will be showing what it's for shortly when I talk about translation. But messenger RNA, let's move back to that actually. Messenger RNA, it carries a message. So, during a process called transcription, which I will talk about shortly, the DNA opens up the same as it would when the DNA is being replicated, but it only opens in a region called a promoter. Every gene has a promoter in front of it. A particular region will open in front of a particular promoter, and an enzyme called RNA polymerase will bind to that promoter and will walk along just that gene, just that one gene, so maybe a few thousand bases, and it will make a single strand of messenger RNA, which is a mirror image of that gene. Because it's RNA, obviously, it will contain uracil instead of thymine. So you might have adenine, uracil, guanine, cytosine, all the way along. That is the instructions to make a protein. So messenger RNA carries the instructions to make a protein from the DNA to the replication machinery where you make the proteins. So there are lots of messenger RNAs in your cells for all sorts of different genes. They're constantly being transcribed, which basically means they're being made. A gene opens up at the promoter, RNA polymerase walks along, makes a single strand of messenger RNA, then that is released, it goes along to the machinery in the cell that makes proteins, machinery known as ribosomes, and a protein is made. Okay, so moving on. tRNA. What is tRNA? Well, when I talk about translation, I'll be showing you that at the site of translation where we make proteins, there's a machine called a ribosome. That's where we make the proteins in the cytoplasm of the cell. Now, at the site of the protein synthesis, the ribosome, you need to bring together lots of amino acids. Proteins are made of amino acids stuck together. Now, the messenger RNA tells the ribosome which amino acids to put together, But those amino acids need to be brought to the ribosome, and they are brought by transfer RNA. So this is made of RNA. It's a piece of curled up RNA, and it has attached to it an amino acid, the building blocks of protein. There are lots of different tRNAs, transfer RNAs, that transfer different amino acids to the ribosome. So the amino acid serine as a tRNA, the amino acid proline as a tRNA, etc., tyrosine. So tRNAs carry different amino acids to the ribosome, where they can be assembled according to the instructions of the messenger RNA to make a protein. I will explain what this anticodon does shortly. Very important region of the tRNA. Then rRNA, ribosomal RNA. Now, the ribosomes are the machines that make proteins, but these machines are actually made of RNA, a special type of RNA called ribosomal RNA that's wrapped and coiled up to make these machines. Now, ribosomes come in two pieces, a large subunit and a small subunit. The large and small subunit comes together to make a ribosome. Now, we have two different, actually, I won't talk really at this point about the different sizes of ribosomes. They're slightly different sizes in bacteria than they are in eukaryotes, but in both there is a large subunit and there is a small subunit. They come together to make a ribosome. The messenger RNA binds to them. It is read by the ribosome. tRNAs come along, bring amino acids, and they are assembled according to the instructions of the messenger RNA to make a specific protein, such as insulin or collagen or keratin or whatever. Okay, so moving back. Okay, so we've covered that. Now let's talk about DNA makes RNA. So how do we get the messenger RNA from the DNA? Well, I've sort of told you already, but just to summarize, the process is known as transcription. We transcribe the DNA code into messenger RNA. We create a messenger RNA which carries the instructions from a particular gene to the ribosome to make a particular protein. Transcription involves enzymes called RNA polymerases. In bacteria, these are usually called transcriptase. They interact with specific regions of DNA called promoters, which are usually just in front of the coding regions. Promoters have a lot of A's and T's. The reason for this is that they're easy to open. You see, adenine and thymine are bound together with 2 hydrogen bonds, whereas guanines and cytosines are bound together with 3 hydrogen bonds. Therefore, it's easy to open up A's and T's or melt them than it is to open C's and G's. So promoters tend to have a lot of A's and T's because obviously the DNA needs to be opened up so that it can be copied into mRNA. Okay, the first step is making your pre-mRNA. Oh, let's zoom in. Sometimes it zooms out again. Okay, so pre-mRNA. So this is a double-stranded piece of DNA. Little bubble opens up at the promoter where the RNA polymerase binds. And then the RNA polymerase walks along the bubble until it gets to the end of that gene, and it makes a piece of messenger RNA, which is then released, and the bubble closes. This piece of messenger RNA can then move along, still in the nucleus at the moment, and go through a nuclear pore to leave the nucleus and enter the liquid of the cell, the cytoplasm, where it can find a ribosome to give the instructions to make a protein. But we have not really made messenger RNA yet. We have made something called pre-mRNA first. So this piece of single strand that we have is known as pre-mRNA. And it consists of sections known as introns and sections known as exons. Introns and exons. I should say that this is only in eukaryotic cells. In prokaryotic cells, there are just exons, no introns. They're just regions of the pre-mRNA. Now, the introns are parts of the messenger RNA that are not going to be used to carry the instructions to make a protein. They used to be called junk bases, but they're not junk really. I won't explain exactly what they are used for right now, but in many cases they aren't junk. The point is they don't help the ribosome to make proteins. Only the exons do that. The exons carry the instructions, the introns don't. Imagine along this piece of pre-mRNA you have intron, exon, intron, exon, intron, exon. The introns are junk, the exons are good. They carry the instructions to make a protein. So what needs to happen is this pre-mRNA needs to be turned into mRNA. This happens by snipping out the introns and sewing the exons together. Once again, these introns and exons are just sequences of bases. The introns are not needed, the exons are. So the introns are chopped out, the exons are sewed together. You now have proper mRNA, which can leave the nucleus through a nuclear pore. Pre-mRNA cannot. So genes are usually not the main part of the eukaryotic chromosome. In human DNA, only 3% is coding, the genes. And even then, most coding regions contain a mixture of actual coding exons and non-coding introns. I'm not going to talk right now about what the rest of the DNA does, but a lot of it is involved in regulation and structure and various other things. Messenger RNA. This is how the, once again, zoom, zoom, zoom. This is how the pre-mRNA becomes mRNA. Here's an exon, here's an intron, here's an exon. An enzyme comes along and it recognizes the boundaries between introns and exons. And what it does is it snips. It snips the intron at one end, snips it at another. It is released as a intron lariat, which are then digested in the cell. And then the exons are sewed together. And this happens all along the pre-mRNA. Introns are removed as lariat loops. Exons are sewed together. You now have mRNA, which can now leave the nucleus of the cell through the nuclear pores. There's an animation about that later. It can go to the ribosomes and proteins can be made. Okay, so moving on. We have now basically just covered transcription. Now we need to cover translation. How do we make the proteins? The process of translation. That's how we make proteins, translation. So the process is called translation. It happens in structures called ribosomes, as I've mentioned several times now. The RNA message is written in a 3-letter base code. So each 3 bases along the messenger RNA are known as a codon. A codon. And each one is the instruction to add a particular amino acid to the growing protein at the ribosome. So maybe 3 bases together carry the instructions to make to add a proline, another 3 methionine, another 3 serine. So these groups of 3 are the codons. The collection of all these codons is known as our genetic code. And this is how it works. Okay, let's just zoom out ever so slightly. Whoops. Just want us to get this right. Okay, we can all see what's going on here. Excuse me 1 second, the cat needs to leave the room. I'm not really standing in front of a college in Cambridge. There you go. The cats will probably demand to re-entry into the room shortly. Okay, I am living in Cambridge, by the way. I'm not studying in front of a college right now, but I am living in Cambridge. Beautiful city, by the way, beautiful city. Okay, right, so this is known as translation. This is how we go from the instructions of the messenger RNA to generating a protein at the ribosome. So this big grey thing here is a ribosome. These little curly things here are the tRNAs. Remember the transfer RNAs that carry amino acids. This is a methionine amino acid, a valine amino acid. And you can see the sequences of bases along the messenger RNA. Each group of 3 is a codon, the instruction to add a particular amino acid to a growing protein. So, first thing that happens is the messenger RNA binds to a ribosome. Ribosomes have a couple of slots in them that can fit very neatly tRNAs. tRNAs have an anticodon at the bottom of them, an anticodon. Let me just show you that again, actually, so we can kind of, we need to go out a couple of levels to sort of see that. And if we zoom back in here, you can see a tRNA. At the bottom, there is an anticodon. An anticodon is just like a codon, but it's the opposite sequence. So imagine you had a codon that was CCC. Then the anticodon for that would be GGG. GGG binds with CCC. Remember, A's bind with T's, C's bind with G's, except with RNA, and these things are made of RNA. Instead of a T, you have a U, so it's U's binding with A's. Okay, so back again, and back again, and back one more time. And then we'll move forward and take another look. So what's really going on here? So the ribosome is bound to the messenger RNA. And a tRNA comes along. Now there's a sequence where the messenger RNA starts. And it's always a methionine sequence, the sequence of the amino acid methionine. A tRNA will come along and it will slot into the ribosome. And if it's the correct tRNA with the anticodon for methionine, so methionine is AUG, so the anticodon will be UAC. If methionine comes along with the right sequence, sorry, the tRNA comes along with the right sequence, the methionine tRNA, it will bind and stick. If the wrong tRNA comes along, then its anticodon won't fit the codon, so they won't bind and it will just be ejected from the ribosome. But there are many tRNAs floating around the cell, so very quickly the correct tRNA will bind, and when it binds, it sticks in the slot next to it. There is room for another tRNA to come along. Now, that could be any tRNA, whatever this codon is the sequence for. Several tRNAs will try and fit, but they will have the wrong codon because they carry the wrong amino acid. Sorry, the wrong anticodon, because they will carry the wrong amino acid. But eventually, the tRNA with the correct anticodon will bind to the codon here, and they will both stick. There is an enzyme present here known as peptidyl transferase, and what that will do is it will cause the 2 amino acids, the one from each tRNA, to bind together to make a dipeptide, 2 amino acids stuck together, at which point the mRNA will move along through the ribosome 1 codon. It's moved along 1. When it moves along 1 with the tRNA still there, that first tRNA will be kicked out and the second tRNA will move from the second slot to the first slot, but it will keep the amino acid bound to it because there's a peptide bond. This frees up the second slot for another tRNA to come along, which it will do very rapidly, and it will fit if its anticodon fits the next codon, and it will bind. Its amino acid will stick to the 2 that are already there. Everything will move along 1 more codon. The second tRNA will be kicked out, leaving its amino acid behind, and this will continue and continue and continue all the way along the messenger RNA, growing this big, long peptide chain, a polypeptide. Eventually, you get to a codon known as a stop codon. That isn't a codon that binds with a tRNA. It's a codon that tells the ribosome this is the end of the message. You have made the protein now. And at the stop codon, the messenger RNA will be released from the ribosome, and so will this big, long polypeptide chain, which might be thousands of amino acids long. It will then fold up, There are certain enzymes in the cell known as chaperone proteins, which help it to fold up into the right shape, and you've made a protein. That is known as translation. Simple. Remember, there is a pause and a rewind if you want to watch this again. Okay, so moving back, let's summarize. By the way, there is an animation coming along, quite a good one actually. I do advise waiting for that. The central dogma of molecular biology. Francis Crick first proposed this in 1958, and it was slightly reworded in 1970. DNA makes DNA, and DNA makes RNA, which makes protein. DNA makes DNA is replication, DNA makes RNA is transcription, RNA makes protein is translation, and here's a summary. Now, before I play the animation, very good animation, I'd just like to say thank you for watching. I'm Dr. Mike Darcy. Here is my Twitter handle. I'm very controversial on Twitter, I do warn you. And now what I think I'm going to do is play you a nice animation that shows you some of these processes actually occurring with subtitles. This, by the way, I should credit it. It's from YTV, created for the Victoria and Albert Museum exhibition. The Future Starts Here, back in 2018. So here we go. Oh, let's start from the beginning. Dum, 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 dum. It picked up where we were last. Here we go. And enjoy. I know I'm going to. This is obviously how eukaryotic cells organize their DNA. These are the nucleosomes. Prokaryotic cells do not contain nucleosomes. They just have the circular piece of DNA. Here we're seeing the replicated DNA, the replicated chromosomes being pulled left and right to opposite ends of the cell before the cell divides in two in a process called mitosis. And here are all the enzymes involved in DNA replication. Remember the polymerase, DNA polymerase, the gyrase, the helicase, all interacting like a fine-tuned musical instrument. So now we're seeing a particular gene being transcribed, being activated. Vitamins are important for some of these processes too, as you could just see from vitamin D. So this is gene transcription. So the RNA polymerase moving along and making a piece of messenger RNA. DNA makes RNA. This occurs incredibly quickly, which it has to, because we have so much DNA in our cells. 25,000 genes, almost 2 meters of DNA. Epigenetics is just the method by which certain genes are switched on or off. I have given a lecture on this. I do have a lecture on epigenetics. Within our cells, there are 25,000 genes, but in most cells, most genes and many genes are not switched on. You don't want a skin cell gene to be switched on, for example, in the heart cell or a neuron. So epigenetics is a method by which certain genes can be permanently deactivated in a cell type or left switched on. Certain genes are tagged to be inactive. When a gene is switched off, it becomes tagged by things called methyl groups, and acetylation is involved too. And this allows certain genes to be condensed, hypercondensed, so that the RNA polymerase cannot get to them and those genes will be switched off. It's very good animation. Okay, thank you very much for watching. And I look forward to speaking with you all again very, very soon. Take care.